I'd just like to welcome everyone to our Café Scientifique tonight on the topic of heart genetics predicting the future. Just to run through, this is a Café Scientifique's been running for, this is the third one we've run, and the idea of these is it's an informal forum for researchers and members of the public to get together and discuss new research and interesting scientific issues. I'd just like to introduce the two speakers tonight. We have Stuart Cook, who is Professor of Molecular Cardiology at Imperial College London, and also Director of the Genetics and Genomics at the Royal Brompton Cardiovascular Biomedical Research Unit, and Tessa Homfray, who's Genetics Consultant at St George's and also at the Royal Brompton Hospital as part of the team in our new Inherited Cardiac Conditions Clinic.
Thanks, Julia. So, good evening, everybody. Thank you for coming. I have a brief here which we will try and cover in 10 minutes, and which covers how genes may play a role in your heart health, how genes influence your chance of developing certain inherited heart conditions, personalized care, and some interesting questions, personally, socially, and ethically. So if we can do that in 10 minutes, good luck. But we'll do our best to address some of those issues. So to introduce where we're coming from here in the genetics laboratory at the Brompton as to what we do, what we're trying to do is use the capabilities and the throughput of the new sequencing technologies to address questions that we couldn't previously address. So we've been very fortunate as a scientist to live in this age when there has been a revolution in our sequencing technologies. And so what has happened is, in years gone by, to sequence a single gene would cost you a lot of money, let's say £1,000, and it would take maybe 3 months to do that for a reasonable sized gene, which might cause you to have an inherited condition of your heart. What we can do now, and these numbers change yearly, they halve yearly, is we can sequence not just one gene, we can sequence, let's say, 200 genes for half the price in about a third of the time or less. So what this really opens up for us in the diseases that we're interested in this hospital and our patients here in this hospital is the ability to comprehensively address the genetic underpinnings of their disease, which we couldn't do previously. So by way of example, one area that we're very interested in, and we have a lot of patients coming through the MRI unit here, is a condition called dilated cardiomyopathy. So dilated cardiomyopathy is when the heart muscle is thinned and weakened and the heart becomes big and baggy and is not able to pump out enough blood to supply the needs of the body. Now, over the years, we've come to understand that this disease is caused by multiple genes, not by a single gene, maybe up to 50 different genes. And the problem that raises is that we have not traditionally been able to sequence all those genes. We just couldn't physically do it, we couldn't afford it, and it took too long. But now with these new technologies, the landscape is changing, and we can use this next generation sequencing to assay all of those genes at the same time. And that has led to some publications. We had a publication last year in the New England Journal where we showed that mutations in this gene titin, the biggest gene in the human body, which is encoded by a DNA which is 100,000 letters long, so it's an extremely long gene, that it is the commonest cause of this dilated cardiomyopathy that runs in families. It does also open doors to problems, and the problems being that we don't know what to do with all the data that we have. So only some of that data can we point a finger and say that sequence variation there is important because it does a certain thing to the protein and it runs in a family. We also get thousands of other variants that we currently don't know what to do with. And so there are ways to address this, and what we're trying to do locally here and in conjunction with colleagues both in the UK and abroad is to build very rich databases with as much information as we can stack into them with the patients as carefully phenotyped as we can to help us interpret what these different variations mean. Because in terms of using that data meaningfully for the patient, either to confirm the diagnosis or to then go on to the family and screen the family and interpret what those variants mean, we need to have that data, which we don't currently have at our fingertips. So that's a very big area. Another area that we're extremely interested in pushing forward, apart from the research side, which is finding what the variants do and how to interpret them, is on the diagnostic side. So one thing that we in the research community and the government are very keen on doing is taking our clinical research, which is enabled by our patients who volunteer to take part, which we couldn't do without them, is taking that research and bringing it back into the clinic. So we want to take the data that we generate, which we need to inform what variants do. We want to then take that back in a package that we can use in the clinical environment, which means turning our research work, which is not as well controlled as you might have a clinical environment, into an assay that we can actually use clinically, and we can then return results to our patients. And so we're very lucky to have been awarded a grant from the Wellcome Trust and the National Institute for Health Research for £1.8 million. And what they are asking us to do is they see the value of what we've done in the research arena, and they are asking us now to turn that into clinical tests for our patients. So that we can, when Tessa sees a patient in the clinic and she would like to add on that added level of genetic information, and to be clear, genetic information is just another test. In medicine, we don't ever just do one test. We don't just do a blood test. We don't just do an X-ray. We don't just do genetics. We use all that information together. So it's added value. But if Tessa wants to do that test, she can order it. We'll have a system where it gets processed through the laboratory, and a report will come back to the clinician, who can then an interpreted report, which can then be used for the patient's benefit. So a big push of what we're trying to do is take our research data and bring it back into the clinic for the patient benefit. I guess we may touch on this today. One aspect that we do within this research, and the community does in general, is when we invite a patient to take part in a study, we give them the option of returning data which we can interpret back to the patient. So although we point out and go great lengths to say it may take years before we get any answers, the experiment may not be done for a decade, and when we do get the data we may not know what to do with it, but if we do get something that's useful and you want to know, then we will contact you and give you that information back after counselling. And I think what has happened with accumulation of a lot of genetic data over the last decade is this data has been accumulated but has not been fed back always. But I think there's a move within our community that we feel that if people wish to have that information should be given back if it's useful. And so this is something that we do here, and is enabled by having Tessa, that we are able to give this information back to the patients meaningfully. What else do I want to say? Sudden death. So an area of great complexity, genetic complexity, is the area of sudden death, which is catastrophic in the families where it happens, often affecting young individuals, children and young adults. Not uncommonly, that will have a genetic basis. Not always, but not uncommonly. And again, a problem that we've had in screening for this is the complexity. Too many genes, how do we assay them all? And what we hope to do now is, in collaboration with other people, is to deliver a gene panel, a diagnostic test where we can screen for all genes that might cause sudden death. So this can be used in an area which is very much growing in the UK now, which is called molecular autopsy. So quite often in these cases they may go to the pathologist in these terrible cases, and the pathologist will not find anything at the level of the organ. There won't be anything to see. But what we can do now is layer on top of that the genetic information, which will be informative in a number of cases, and that may then help. It won't obviously help the unfortunate patient who's died, but it may help screening with the family and family members. So that's another area that we're particularly interested in. So I think that's about all I had to say, really. Tessa?
How do I work this? Yeah, I've got it. So I'm very clinical, so I'm not spent my time in the lab. And I get asked 4 questions, really, by patients. First of all, is there any point? I was born with this problem. Is it just all about whatever is there, is it all inevitable? Well, actually, of course, it's not inevitable. There are things that we can do about it. We may not be able to change our genes, but we certainly can alter how they are going to function. So I do believe that we are able to develop many better treatments for these diseases by knowing the exact cause, and maybe we can explore that a little bit further. Another thing that patients very much ask me is, what is the risk to my relatives? And of course, without knowing the underlying cause, it is absolutely impossible to actually look at that further. So by actually getting back to the basic molecular diagnosis can at least give us some information of whether somebody is actually at risk. One of the things, though, that we do notice with these diseases, that there will be a large number of family members who potentially are at risk, but very few who actually become affected. And this is something that we call penetrance in a gene. So even though they may carry an abnormality, their actual risk of developing the disease may be, in fact, really quite limited. And of course, we need to find out why that is. Is there second genes that are involved, which there's less that we can do about? Or is it that there are environmental triggers or treatments that we actually can potentially do something about? And I think we've got good evidence for other parts of medicine that there are things that we can do to prevent these diseases manifesting. There are specific treatments for specific genetic disorders. There's a condition called Fabry's disease, for instance, and this is a disorder that causes hypertrophic cardiomyopathy, and there's a specific enzyme replacement for this. So diagnosing it, you can actually give an absolute specific treatment that will fundamentally prevent you getting the disease. So that, of course, is incredibly exciting. One of the other things that we talk about is, can these things skip generations? Because up until now, what we've always done is clinically screen first-degree relatives being parents, children, brothers and sisters. And of course, if something has this reduced penetrance, of course, it can skip generations. The disease can skip generation, the gene does not skip generations. So by offering a simple blood test to someone who is clinically unaffected, we can then say whether their children are at risk or not. And that is a huge advantage of who we need to then screen. We can discharge large numbers of patients because they are at no increased risk. And for these patients living with this cloud, particularly of the genes that can cause sudden death, it is very, very, very tricky. Now, of course, for 50% often, they remain with that risk, but at least we can offer often very effective treatment. There is also preventative treatment. We can avoid them taking certain medications that actually might prevent them developing the disease or having manifestations of the disease. So again, a fairly simple intervention. There are certain antibiotics, antihistamines that you shouldn't take if you have something called long QT syndrome. So that's pretty easy because there are always alternatives that are available. I think the other thing that is, I often get asked in the clinic is there any disadvantage of testing? And the one that I discuss most is the risk in the insurance industry. Now, in general genetics, and I'm involved not just with cardiac genetics, insurance companies cannot ask you the result of your genetic test. What they are able to do is ask your family history. And therefore, actually, what in fact makes a difference is if you've got a positive family history. So if your father died, say, age 40 of a hypertrophic cardiomyopathy, you actually have to declare that result. If, for instance, though, you have had a genetic test which shows you do not carry the gene that has been identified in the family, you can go to the insurance company and say, actually, I don't have what has caused my father's death. I don't need to be weighted. So actually, in that case, it is an advantage. Of course, the other side is when you do carry the gene, but then we undertake clinical testing, and it is only if they have clinical abnormalities that they need to declare that to insurance companies. There are a certain number of career issues, though, and I think with these genes, there are potential issues with things like the airline industry, and certainly I am not quite certain where we stand as far as this. I've actually taken it up with the Civil Aviation Authority, but if you know that you shouldn't become an airline pilot, it's probably better to know that beforehand than actually become your airline pilot and then said you can't actually do that job. So although it is worrying that there may be career limitations, I still think you are probably better knowing before you've actually gone on that pathway. And that certainly is the same for some of the sporting conditions, that it may be completely unsuitable to you to go and be an Olympian if you are at risk of developing one of these diseases. Those patients are all clinically screened anyway nowadays, so they may well be stopped at that stage. So again, they will have been over-trained, and therefore it's a huge disadvantage. I think there are some people who live their lives with this weight on their shoulders, and I can fully understand that. And if you have a child with one of these conditions, how do you let them out of your sight? And I always think, how would I be as a parent under those circumstances? But I think the great thing is that people get used to this information, and if they only need to worry about certain of their children, I think I'm amazed how well people do cope, but I can see that being a problem. But bearing in mind they were already worried, I'm not sure how much difference the genetic results actually are as a disadvantage. So I think there are cautions, and people need to know what they are actually being tested for. But I think with the possibility of specific treatments, of discharging a large number of patients from the clinic, and the possibility of interventions actually preventing anything, I personally think we have got some very exciting new tests and possible new treatments on the horizon. So I think that's all I have to say at the moment, and I think it's now over to you.
On again? Oh, yes. If you just want to raise your hand, and I'll bring the mic.
Hello. I have dilated cardiomyopathy, and since about 2000, when I developed the symptoms, and I've been well looked after by King's, in fact. But never once have they suggested any genetic testing of my sons, of which I have three, two in America and one here, and I now have three grandchildren through them. So am I being, in your view, sort of derelict in my duty, sort of antisocial or a bad father, in not because of the low penetrance that you referred to? I mean, you can't afford to sort of underestimate the effect if when I see them and I say, look, the heart disease I've got, and they all know I've got a heart problem, but they're not interested in really the details of it. And I haven't deliberately kept it from them, but they just haven't asked. They aren't airline pilots, or strenuous people in particularly. So I get the feeling that you probably think they should be tested immediately, but what do you think about that?
Maybe I'll start. So thanks for that question. So I think there's many layers of an answer to that question. And first of all, I would ask, have they been screened at all clinically? Have they had ECGs, echoes, or imaging? Nothing at all, nothing. So obviously, lots of this comes to, exactly as you've said, to penetrance and also to expressivity. So expressivity being, if you have the disease, how bad do you have the disease? And penetrance, if you have the gene, do you get the disease at all? And so clinically, what's most important in this situation would be, in my opinion, the imaging analysis of their hearts. If they have a functioning normal heart, then, and they're in their middle age, then things are looking pretty good. Coming back to the genetic testing, and one reason why genetic testing hasn't been used routinely in dilated cardiomyopathy is because it hasn't had a good yield traditionally. So if you were to sequence all the known genes and you had all the money in the world, you'd be lucky to pick up a genetic cause in 25% of people. Lucky, with the wind behind you. With the new gene that we've discovered, we can double that. We believe we can double that. So we believe we can now get you up to sort of 50%. So one reason for not doing the testing was the fact that you just didn't pick, you couldn't screen enough, and your yield was much lower, and you were better off doing the clinical phenotyping, which is more important for outcomes anyway.
Yes, I think my always concern is what risk are you leaving them with? And I think for dilated cardiomyopathy, in people who haven't had major rhythm disturbance, the risk of sudden death, which is to a certain extent the most frightening risk we have for these diseases, is actually low. And therefore, I am not quite as anxious as I am with people, say, with hypertrophic cardiomyopathy, who we know that teenage lads, drop dead on the football pitch. And therefore, I think the disease that we are dealing with certainly influences how much we actually worry about it. Now, I would agree with Stuart. Dilated cardiomyopathy has been genetically very, very poor yield. I would like to sneakily look at your children's hearts and, look at their echoes. And again, as Stuart said, if their echoes and ECGs are all right. And we know not all dilated cardiomyopathy is genetic. Now, whether there is a genetic predisposition in people who get viral myocarditis and things like that, there probably is. But I think from the dilated side, it's probably the least of the issues over the other ones.
Questions?
Hi. I've come to this meeting because it was about cardiomyopathy. Now, cardiomyopathy has got so many different variants, types of cardiomyopathy, but I have, over the last few years, come to realise that research has become quite a lottery. I mean, I have HCM, and it's coming towards end stage. I've got bivent. I had my blood taken in 2007 for genetic testing, and it's still in the freezer. I've been to an information day a couple of years ago, and a certain professor held his hands up and says, yes, DCM is moving forward in research, but I'm sorry to say that HCM has been put on the back burner. Now, with all these fundings and genetic testing, as HCM sufferers, we feel that we're just plodding along without the support that DCM patients have. Hypertrophic cardiomyopathy, which years ago, because I was diagnosed late 70s, it was HOCM, and that was the main recognisable cardiomyopathy, but we've been left behind.
So, I'm sorry you feel that like that. Did I just ask, with the blood sample that was taken for the genetics, was that taken for for research or for clinical testing?
I was just asked if they could take my blood to see what gene I have, and every time I go to clinic, it's in the freezer.
Okay, so I mean, there's a very big distinction, because obviously, and we try really to make this explicit, because if it is taken for research, it is possible that it will not be analysed for a decade. It's possible that we may not do that. It would be biobanked, is what we call it. If it was taken for a clinical purpose and you didn't get the result back, that would seem very odd to me, indeed, because that is turned round. It does take 6 months, but it is done and it is turned round. With regard to the genetics of hypertrophic cardiomyopathy, that has been very well studied and very well described, and you're correct in saying that we have not made many recent discoveries in that area. There have been some, but most of it had already been described in the late 80s and early 90s. So the newer discoveries are coming around based in dilated cardiomyopathy. You're absolutely correct. But it's not that the HCM has been left behind. It's that we can explain 80-90% of cases now using the genes that we already know. So I don't know if that explains much, but that's kind of hopefully of some use.
I think it's absolutely true that there are certain places where you don't get genetic testing and others where you do. So there is absolutely no doubt, and I know a number of hospitals where they just freeze them. We actually do test anyone who has got good going HCM. We would always genetically test. I think to say nothing has happened isn't quite true. 2 years ago, we were able, because of this new old technology, which was all we had, to test for 5 genes. And we are now routinely testing for 14, and we are soon going to go up for testing to sort of 50. So although there isn't the sort of research that you're talking about that is taking off in DCM, we're now going to have the technology that we can look at much, much, much more. So even though there is progress, it just always seems very slow when you're the patient, doesn't it? It's never quite quick enough for you. And I think that's absolutely true. But it is moving, and I think that everybody should get tested. And certainly we do here.
Just add one more thing to that is, with the journey of research, from the basic discovery of the genes, which takes years, you then have decades of research to chase up the mechanisms of those diseases. And when you figure that out, you can then develop drugs to treat those mechanisms. So with HCM, the discovery was done a while ago, and we are making incremental advances on our knowledge in the area. But what has happened is the transition from the discovery into mechanisms. And so now there's lots of studies being done in all sorts of different model systems where people are now eking out mechanisms, pointing towards fibroblast activity, which is now informing new treatments for HCM, which obviously, in your personal case, is coming a little bit late. But it's made that transition from the discovery to the mechanism to the therapies. So actually, HCM is ahead and is doing better because it's further down the route of the scientific discovery and translation.
Thank you very much. I was really interested to hear about the sudden adult death research. It sounds fascinating. Someone very close to me died of that a number of years ago. I have a couple of questions to both of you, really. So first of all, do you think it's likely within the next 10 or 15 years that there will be a genetic test or possible to screen people to be predisposed to sudden adult death syndrome? And the second part of that question is a more clinical one, really, which is what would be the advice if you were screened and, yes, you are at risk of sudden adult death syndrome. And then the sort of the moral question is something like ignorance is bliss relevant in those kind of cases if it is, in some cases, there is no clinical intervention you can make? So there's sort of 3 questions there, really, and I'd be interested in hearing about any of those.
Yeah, I think it is very difficult. Whenever you are considering screening for any disease, there is a fallout. And I think we have to be hugely, hugely careful. And if you look at clinical screening, and there are people in CRI and other organizations who are offering ECGs, routine ECGs, to sort of teenagers, and they've been going around posh public schools, because obviously you're more important if you've been to a posh public school, so you must be screened. Anyway, they've gone around these public schools offering ECGs, and they find slightly abnormal ECGs. These kids then come into our clinics having then, with slightly iffy results. We stop their sports, we stop their all their sort of normal life events. It causes havoc in the family. So I think we have to be very, very careful about mass screening. Because of what are we going to do about it. So I absolutely agree with you, that sort of moral dilemma that you have. I think once there has been a major event within a family, and that doesn't have to be sudden death, that can be syncope. So you can have someone who's fainted, or they've been resuscitated, or something, equally successful. And therefore, and there is then, we make a diagnosis, and therefore screening relatives seems to me at that stage absolutely reasonable. And actually, certainly as a parent, if you've seen one of your children go through one of those incidences, the thought of not having your other children tested, I think, would be a pretty tough call. But you've got the side effects of the medication that we start. And beta blockers, which I'm sure a lot of you know about, are the mainstay of treatment for most of these diseases. A lot of people have no side effects at all, but there are certain people who feel like complete zombies on them, so they go through life as a zombie, which, if your risk was actually very low and you haven't come across it, I'm not sure that many people would actually take it. And these people, they come and get their prescriptions, but when you go through people's houses, there are amount of prescriptions that are not actually taken. And so people vote with their feet. So it is an absolute tragedy when someone dies suddenly. But to screen a whole population and treat people who are never going to run into any difficulty, and unless you've got a treatment that has got no side effects at all, I think it's a very difficult idea. And I'm very ambivalent about screening.
Hi. I take the point completely that you don't want to screen the whole population of youngsters. Not cost effective, and you pick up slight things that would worry people. But when you've had an event in the family, then it's completely different. People are motivated to be taking whatever medication. So my question is, once you've had an event in a teenage population, whether it's long QT, I think you mentioned, hypertrophic cardiomyopathy, Brugada syndrome, whatever it is, and then you do an ECG, you do an echo, you do genetic testing on the at-risk siblings, does that exercise save lives?
Does it save lives? I think the real difficulty, you don't know. We still don't have a really good indication of what would have happened with those family members if they hadn't been on treatment. Now, there is a condition called catecholaminergic polymorphic ventricular tachycardia, or CPVT, which is a little bit easier than that. That's a rather long name. And there's very good evidence with that condition, where there is, 40% of people have had serious events by the age of sort of 35. And in those, looking actually at intervention seems to make a major difference, because there is the high penetrance. And the difficulty we have is this penetrance issue. And now with hypertrophic cardiomyopathy, it is easier to look at things because you have an obvious phenotype for that. So it is easier to look at interventions with that. But with the long QT, it is difficult to know exactly how many lives that we have actually saved. So I think we make a difference.
One comment on that specific example. If you have an event in a family and you have an a priori and you know that the probably background genetic burden in that family is going to influence the trait, and if you have a causative variant and you're convinced that this is the variant causing the disease, finding that variant in affected family members may not save their lives, but it will influence your management. But one point that Tessa's pointed up before, I think is a very nice way of putting it, is those who don't have that disease, that mutation, you can not save their lives, but you can save their lifestyles, because you can discharge them to health. And that's a very big thing.
I've got a question. Can this be diagnosed in the womb before a child is born? Can you test in the womb?
We certainly can test in the womb. How many times? My other major interest is prenatal diagnosis, so actually I'm a very reasonable person to ask for that. I've got to say, I don't really encourage it. Now, I'm not living with these diseases, and maybe I should be saying these diseases are so terrible that one shouldn't have children with these conditions. But, you know, you look at completely normal people wandering around, you know, again, the penetrance is not very high in a lot of them. To undertake termination of pregnancy for these sort of conditions, I feel uncomfortable. It is a personal choice because I'm not living with the disease, and so if patients say to me this is what they wanted to do, I would of course agree that it is their rights, but it's not something that we do very often. There is something called pre-implantation genetic diagnosis, where you can select embryos that are unaffected. So if that is an ethically better option for people, then we can do it without the sort of problem of termination of pregnancy. But we have to know the genetics before we actually go into it.
Thank you.
Can I just make a comment on the pre-implantation genetic diagnostic? So in some very severe forms of disease, for instance, very severe lamin gene cardiomyopathies, which are catastrophic in families, if you do, and this does happen, you generate the in vitro embryos, they are screened, and then the negative ones are implanted. And then that prevents the passing on of the disease within the family at the cost of throwing some embryos away.
You mentioned that the gene is carried through every generation, but the disease doesn't necessarily manifest itself in every generation. By which mechanism does that happen? How is it provoked? And how certain can you be that it's not carried through every generation if it doesn't manifest itself? Obviously, the gentleman over there, for instance, I think he said the year 2000 it happened, some event, and unless a serious event happens, how can you be sure it's not carried through every person carrying that through the generations?
Well, if you look at people who carry the gene, okay, so the gene is in every single cell in your body. It may only affect your heart. So you can tell if that patient carries the gene, and you can then screen them clinically, depending on what disease you are actually looking at, to see if they've got any clinical manifestations. Now, we absolutely know with some of these arrhythmias, they may have absolutely no clinical manifestations at all anyway, and the first presentation would be a catastrophic collapse. So I agree that you don't know that they are never going to develop a problem, and that's why we land up treating people who may never ever land up with a problem. It is different for the cardiomyopathies, where there is a good clinical investigations that can tell us if you've got a thickened heart. So we know with a number of people who carry genes, abnormal genes in the hypertrophic cardiomyopathies, that they never develop anything at all. They have been having regular screening, but they carry the genetic abnormality that has caused a much more major effect in other family members. So we can tell that very easily. And you only treat if you've got clinical symptoms or clinical signs. Whereas with the arrhythmias, it's much, much more difficult. So we do treat people who appear to have absolutely no problems at all. So that is the difficulty.
You can't sort of estimate what kind of event would provoke a manifestation.
Well, with an arrhythmia, it is a faint, a collapse, a cardiac arrest. Those are the normal ones that we actually... A lot of people used to be diagnosed with epilepsy, and in fact these were cardiac collapses. So that's what we are really looking at, and sometimes when we're talking about screening, as we were over there, what I really like to be able to see is when people do have faints, that they are properly investigated and not just put down to, if you are running a school, your egg and spoon race, and you pass out, it's not normal to pass out in your egg and spoon race. It's not that traumatic, is it? No. And so I think, if that happens, if you have a faint in a swimming pool or near a swimming pool, you need investigation. So I think it's the clinical suspicion that needs to be raised.
Are there degrees of importance? Are some more dangerous events than others?
Yes, absolutely. And we certainly know with things like long QT or that CPVT that I was talking about, that once you've had an event, you are at much, much, much higher risk of a further event than you were before you had your first event. I mean, that all sounds a bit counterintuitive, but so once you've had an event, the chances of having a second event are very high. So you have to treat people at that stage. So picking someone up who's had a faint secondary to this is extremely important, because the intervention then is successful.
Thank you.
Just to mention one thing from the scientific point of view with regard to penetrance, i.e. why some people they definitely have that gene, and you can take it to the extreme case where you have identical twins who have exactly the same DNA, both have the mutation. One of them has very bad disease and the other one doesn't. And the question why is extremely complex, and we could fill this building with researchers across the world who are trying to address that question, because it is very, very complex. It can go back to epigenetics, which is modification of the kind of the sugar molecules stuck on your DNA, not the DNA itself, but what's stuck on it. It could be related to what you eat, it could be related to viruses, it could be with the position that you are in your mother's womb, it could be what she eats, and so it is immensely complex, and we don't know. Sorry? Definitely.
Because we can interfere with the environment. Much more difficult to interfere with the genes.
It does. If you imagine you've got two identical twins, and one has the disease and one doesn't, their genetic load is the same, something else is different, and that potentially, if you could find out what it is, then you can modify it, then you could try to prevent the disease.
I have a couple of questions that you've kind of touched on before. The work that you've done has just been on DCM, and or the work you've talked about today anyway. I potentially had a diagnosis of ARVC a couple of years ago, and attending some of the Cardiomyopathy Association meetings, it seems to me that our understanding of the genes is increasing, but it's obviously becoming more complicated, and the boundaries between the different cardiomyopathies are becoming a bit blurred and so on. And I wonder if you are also looking at saying, well, okay, we're not just going to look at DCM genes, but you have this panel of 50 genes and say, well, all of these are related to some of these diseases, and how do they work together? And I suppose one of the follow-ons from that then is that you also, again, we're saying that we can only pick up a certain percentage of the genes that are related to these diseases at the moment. Presumably there's only a finite number of genes. I mean, is it your hope that at some stage in the future you will be able to say, yes, okay, we can analyse, we know all of the genes that are responsible for these conditions?
So that's a really excellent question. I'd like to offer you a job in my lab.
Spent many years at Imperial already, I don't think.
So no, I think you're absolutely right. I think there's this blurring of the boundaries between these cardiomyopathies. We know it used to be called ARVC, arrhythmogenic right ventricular cardiomyopathy. We then had a form called ALVC, arrhythmogenic left ventricular cardiomyopathy, and now there's a move just to call it AVC, arrhythmogenic cardiomyopathy, because it's merging, and we find the same genes, titin, for instance, which causes dilated cardiomyopathy, can also cause hypertrophic cardiomyopathy. It can also cause ARVC. So I think we're having to re-educate ourselves from what was done in single gene studies over the years and look at it from a higher level and look at all the genes. So it's exactly what you say is exactly what we're doing. So in our panel, when we screen a patient who kindly volunteers and takes part, we screen all the genes. So we screen in every patient who comes through 200 genes. So we screen all ARVC genes, all long QT genes, all TPTV genes, all DCM genes, all ARVC genes. And when you start to look at that data, and we're only just beginning to do this now, we are already seeing patterns that you may have predicted, but are there. So people are getting multiple hits and multiple genes across the pathways, and I think it's going to be really important. So no, I completely agree. With regard to discovery, I think, as I was mentioning in hypertrophic cardiomyopathy, I think most of the discovery has been done. We've still got 10, 15, 20% we need to work on, but a lot of it has been done. I think in dilated cardiomyopathy, we've got a way to go. But we're now bringing the new technologies, again coming to global analysis. So we currently screen the 200 known genes, so we hope to try and clean up as much information as we can get from that. And now we're going to go genome-wide. So we are now looking to screen all genes, as in 25,000 genes that each of us has in our body, in all of our patients, and try and discover what we don't know.
I think the other thing is, if it's not genetic, we're not going to be able to find it. But proving a negative is impossible. And therefore, I think to ever say to someone, we absolutely know that it doesn't exist, you're a pretty arrogant doctor who does that. And I think that is always the problem of totally reassuring someone that nobody else is at risk. Because even when we're screening the genes, there's an awful lot of DNA that is not coding DNA. And therefore, we know that it's not junk like it used to be described as, and there may be sequences within there that are controlling genes. So I think luckily we're going to be in jobs for some years to come, because it's going to take years. So it's proving a negative is really tricky.
Yeah, just a quick question. To clarify what you said, have genome-wide association studies been done on these conditions? And if so, how many thousand patients?
So genome-wide association studies have been done in dilated cardiomyopathy. We published one in 2010 from our group in collaboration with a French group. There's a very big difference here, which is a little bit genetic-y, but I'll try and explain it. So basically, in genome-wide association studies, you look for the effect of common variants, so that's genetic variation that's present in up to 1 in 20 people minimally, but probably more than that, and their effect on disease. Whereas the variants that cause the diseases that we're talking about here occur usually at 1 in 500 for hypertrophic cardiomyopathy, 1 in 2,000 for dilated cardiomyopathy, 1 in 5,000 for CPVT. So you're just not powered to pick those up in genome-wide association studies. They'll only look at the common variants. So what they pick up at is a different type of gene, which is more like a modifier gene, a gene whose expression level might influence the effect of a causative mutation. So what they don't inform on is the rare variants, because if you screen 5,000 people, you might only see it once, and then you don't have any statistics to use. So GWAS doesn't really work. It does kind of work, but it doesn't really work for finding causative disease genes.
Just coming through. It helps colour those patients that you know have definitely got the gene, because if they've got other causative genes, then they're presumably more likely to be worse.
So those are exactly the guys that you want to do. But first of all, you need to know so you need to know their mutation first, that they've got a mutation, and then you can see how that's. And we're doing those experiments, so that's a great experiment to do.
Hi. I'm not sure if it's related to what you're working on, but the News at 10 had news of Alexander Lyons' trial, and that was about heart failure, which is what we're discussing. And I was wondering if is it a very specific group of patients who have just had heart attacks or anything, and also that's a genetic therapy. And can you shed any light on that? Is it relevant to what you're talking about?
So this is the Sirkar Trial. So what we've known for very many years is that in heart failure of all causes, whether you've had a heart attack, you've had a viral illness, or you have a dilated cardiomyopathy, that the ability of the heart muscle to use, store, and recycle calcium is affected. And this has been known for like 2 decades. And so the idea here with this trial, and I'm not working on it, I'm not an expert, but I'm familiar, so I can relay this to you, is basically to simply address the question of, we know there's a problem with the way that the myocyte handles calcium. It's got a problem. We know that Sirkar can make that better. Let's put the Sirkar back in. And it's really as simple as that. We do screen specific, so coming back to the genetics, we believe that knowing the genetic mutation will inform therapy. Which comes to something called stratified medicine, which basically we don't treat everyone with hypertrophic cardiomyopathy the same, we don't treat everyone with dilated cardiomyopathy the same. We use all the information that we have, plus the genetic information, and once we have enough data, we believe that will inform how we treat a patient. And so we have patients who we recruited who have mutations in Sirkar, who have mutations in another gene called phospholamban, which is also controlling the calcium fluxes, and they may be specifically targeted by that kind of therapy. We don't yet have the data. In Titan cardiomyopathy, we think the same thing might be true.
Thank you.
So, having heard this question and answer session, sort of another point is, or sort of a realization has dawned with me that, you know, as so often in life and medicine, it's a trade-off, this testing or screening, forget screening, testing. So, as I said, I have DCM. Now, somebody with DCM, what percentage of their, say, children are found to have the causative gene for DCM and what not? Because it was a good phrase, you can release them to health, but I mean, if you're releasing 10% to health and 90% of whom perhaps only 10% are going to have symptoms at all in their life, you're sentencing 80% to that terrible thing. I mean, we've all, you know, worried about being ill, and we're all terribly psychosomatic. I mean, that's the sort of the trade-off I'm wondering about. Are these percentages discovered and available?
So with the inheritance mode of inheritance of DCM is autosomal dominant primarily, i.e. it runs down in the family and it will affect 50% basically. We come back to the issues of expressivity and penetrance and that we've discussed. I think with regard to knowing or not knowing, I mean, there is this pervasive feeling that people would be better off not to know. But if you already have the possibility of having the disease, then knowing might not be so bad. And actually, there have been large-scale trials to assess this, one done by Eric Topol just a couple of years ago in the New England Journal, involving all sorts of psychoanalytic testing, letting people know or not know, properly controlled and randomized. And actually it turns out that people would actually rather know overall.
Yes, I think for a start you have to identify the mutation in the affected person. So bearing in mind we aren't able to do that in the vast majority of people with DCM, it is not an issue about genetic testing. But yes, the maximum you will ever, there's only a very small group that this doesn't apply for, is going to be 50%. And if you then say the penetrance is, let's say 10%, 10% of 50% are the ones who are going to develop the disease. I mean, that's the sort of figure, but you have to have known what to look for in the first place.
Thank you.
Hi. At the beginning, you referred to personalised medicine, and 5 minutes ago, you referred to stratified medicine. Could you distinguish between the two and their relevance in this topic?
Sure. So, personalised medicine was on my brief. That's why I said it. Stratified medicine is what I believe we can offer to patients in with inherited cardiac conditions, and just to explain why that is. So personalised medicine means that the information made based on that test, and in this case we're looking at genetic tests, specifically informs the management of that one patient. Whereas stratified medicine is the approach that any one patient with that mutation would be treated in the same way. And so that is the big distinction. So personalised is really just that one patient will be influenced. And the place where it really has a role, and it's going to be very, very big, is in cancer. Because there you're looking at the whole genome in that patient, and they may have 6 different mutations which require 4 different chemotherapies, which is particular to that patient. Whereas in inherited cardiac conditions, we are stratifying based on mutation in a gene. To add a caveat to that, I think now that we're getting more and more information on the mutations, it's informing us that all mutations are not the same. And so, for instance, in the case of Titan cardiomyopathy, we now know from a large study which we're writing up as a follow-up to the Titan paper we published last year, that the position of the mutation in the gene matters. So actually, we can, on top of the stratified, you have a mutation in Titan, we can now say, actually, you've got a mutation at this position in Titan, which means you're probably going to do worse, and we need to keep a closer eye on you. So, broadly.
I think the other place that personalised medicine is going to make the massive difference is pharmacogenetics. So different people, we all know certain people who respond to drugs better than others. We know people who have side effects different from others. And those are secondary to genetic effects. So I think that actually that's where the big drive for the personalised medicine is actually going to be. And now some of potentially the dysrhythmias will be much more personalised because the type of mutation may well say which drug is going to suit you better, and then the actual refining that with how much of that drug you potentially need because of your genetic background. So I think personalised medicine is one step further along from the stratification.
So does that mean that all patients will then have to be genetically tested?
Have to. I hope we don't live in that sort of country. So I think there will be a huge drive to test people. Now, if you've got TB, for instance, how you respond to certain drugs is absolutely different depending on how you metabolize them. So it must be better to test those people to get their drug dosage right. Yeah. So at the moment, it's not making a vast difference of genetic testing. I talked about Fabry's disease earlier, and that makes an absolutely vast difference because there is a specific treatment. So if there becomes specific treatment for specific mutations, I think people would be daft not to be tested.
Yeah. And it's the long-term effect of the drugs, because, for example, I was taking Vrapamil since late 70s, and I'm 48 now, and I've got severe fibrosis in both ventricles. So again, if you look at the experiments with mice and verapamil, they showed a lot of fibrosis. Yet my doctors won't say that that was the cause of it. But I've been on that medication long term and a very high dose. But on the other hand, I've led a very active life. You know, I used to sail, whitewater canoeing, the works. If it wasn't for the verapamil, but I've paid the price heavily because now later down the line it's caused all the scarring.
Yeah, I mean, we know with certain chemotherapeutic drugs that they cause a cardiomyopathy, vincristine being it is vincristine, isn't it? Yeah, and it causes a severe cardiomyopathy, but it doesn't in most people. So if you knew which patient was going to be at risk, you'd certainly test them. Now, you've got to save their life first, so it's no point saying, oh, well, the drug doesn't suit you, so you better be dead. I mean, you're better to try and save their lives, but, if there's an alternative, you would want to use it.
How far off with that sort of thing? Is it something that will happen in the next 5 years or so?
Well, they've had a professor of pharmacogenetics who's been appointed in Liverpool. So it is fundamentally possible to actually look at a number of drugs. But there's a huge number of interactions as well. So again, it's not quite, it sounds hugely straightforward the way I describe it, but it's not quite as simple. So it should be relatively near for certain drugs, not for an awful lot of them.
Okay. Another question on that, which was something you talked about earlier when you were talking at the beginning, was that it's a sort of technical question really, but you mentioned that you have at the moment, in some cases, too much data to process. Is that due to the fact that we haven't got big enough computers, or there's not enough hours in the day, or enough staff to do the process? I mean, is this something that will be sort of looked at for future diagnosis and work, so that you'll be able to actually say, we've got a lot of data in the past, we couldn't do it, like it took a long time to process a gene, now we can do it. We can take all this data, we can do a lot more with it, more so than we used to be able to.
Yeah. So just going back to the previous point on the pharmacogenetics, I think one big, just as a brief point on that, one big area which is being driven by the pharmaceutical industries is the fact of companion diagnostics, because most drugs in pharmaceutical industry fail at phase 3 usually, or phase 4 post-marketing, and it's usually due to efficacy, i.e. they don't have a strong enough effect, and they realise that they can actually work out to a degree who will respond and who won't respond based on genetics. And so the future, a very, very big push of the future of drug therapy will be based on genetic stratification. So that's just a point. But coming back to your other point that was about data.
Data crunching.
Yeah. So the machine. So it depends what experiment you do. If you do a whole genome sequence, you get a lot of data. If you do the whole exome, just the genes, not the junk, but not junk DNA, then you get less. Or if you do a targeted resequencing, then you get even less. The biggest amount of data generated in those by the machines is the image files, which are massive. And then the data itself, the sequencing data, depending if it's whole genome, whole exome, or targeted. And shipping that data around is a problem, because we have to ship it around hard disks around the world, because the pipes between the countries and in the internet are not big enough. But what we are able to do now is, because all this data we accumulate in shared databases, we enrich those databases, and then we're able to query those databases just with smaller files. So I think we are addressing it. The files are becoming smaller. We're knowing what we can throw away and what we have to keep. So it is better. My personal feeling, and I think there's strong data to support this, in terms of the diagnostics, we should be using targeted panels and not doing whole exome sequencing. Whole exome sequencing, it's more expensive, it takes longer, it generates a whole load of issues that we have to deal with informatically and ethically. And it's not as good a test as doing the targeted panel, which we create in our labs with much love and care. So, yeah.
Okay.
Yes, I think bioinformatics is a major issue, but every time you have new technology, you create the unknown. And once that unknown becomes known, it becomes much more manageable. So this next generation sequencing is still pretty new at the moment. So we've got this vast amount of data flooding in. We had something called comparative genomic hybridization for looking at chromosome abnormalities. And we had, to start with, the most unbelievable amount of data that we didn't know what to do with, and we kept recalling patients and saying we don't know if this was good or bad, and we were in a frightful muddle as usual. And actually, we've now sorted that out, and we're much, much clearer about what is good and what is bad. So each hump that you go to, but of course, as scientists and doctors, we're always looking for the next new technology. So it'll be there again.
Given what the experience the young lady had over there with her drugs, getting this nasty scarring after I don't know how many years, should we all who are taking heart drugs have some kind of review of what we're taking after a particular number of years? I've got to see, I'm coming back as an outpatient next month, so it's quite pertinent, but I take several drugs for coronary artery disease and several stents to control the dilation of my arteries. But is there a danger they have side effects which I haven't been informed of?
So, I mean, all drugs have side effects. Just to be clear, an aspirin, if it was marketed now, wouldn't get across the counter.
Well, I mean, sort of long-term effects like this.
And so I think, I think within clinical cardiology, so it's moving away from genetics to clinical cardiology. I mean, if we know a drug has a side effect, for instance, it affects your kidneys or it might affect your liver, that would be screened for by your GP or by your physician on a regular basis to make sure that the known side effects were being screened for. What we can't deal with is the unknown side effects. So you should be, if you're on a drug which has a known side effect, was to affect your kidneys, you should be having a blood test to assess that already.
Okay, thank you.
Yeah, I think that's why we like using well-established drugs, because well-established drugs have well-established side effects. So new drugs remain, the long-term side effects will remain unknown.
Is there any place at all in therapy for replacing bad genes with good ones, on a virus or something, as there are in some other conditions?
Gene therapy has been remarkably ineffective. It's, of course, the holy grail of genetics to a certain extent, and certainly when I went into genetics about 20 years ago, it appeared that we were nearly there. We're no nearer there now, really, than we were then. And there are a few diseases for which we have been really pretty effective with, and those have been the immunodeficiency syndromes. They have been good because the body doesn't reject the gene that is re-put in. A lot of the time, the body rejects it, and therefore you immediately mount immune response to the gene that you're trying to give someone. So it's been a major problem. Cystic fibrosis, which of course this hospital has a lot to do with, has been a sad disappointment, because every time we gave the gene to people, it was initially hugely successful, and then the body's own immune system said, we don't like that, and rejected it. So I think that gene therapy for things like cancers, where you've got a limited time where you are going to need treatment, has a high chance of success. But I think actually taking genes out and putting new ones in, we're still, I think conventional medicine still has more to offer.
We take one more question. Is there any more questions? More.
If I go to the local genetics department, say at St George's, can I have a Titan test?
I'm the local geneticist at St George's. What does that say? At the moment, we are doing it from a research point of view, but we are actually confirming them at St George's. We're doing a sort of joint thing. So actually, that's probably the worst lab you could have actually chosen, because it's the one you're going to most quickly be able to actually do it with, but it is not actually available. The only place that I'm aware doing it on a commercial basis is actually in Holland at the moment. I'm sure there's some places in the States, but it hasn't yet been developed in the UK.
Thanks.
Right, I think that's it for questions tonight. So I'd just like to thank everyone for coming along tonight and making it a very interesting discussion, and also to thank Tessa and Stuart for coming along as well. Thanks. I'm just going to hand out a few sheets for evaluation, just to give some feedback to us on this event, and also just to point out that we've got a research open day here on the 20th of May, which is 11:30 to 2 p.m, which is open to the public, so you're welcome to come along and bring your friends and family. And also there's another Café Scientifique, I think there's some sheets over there from the respiratory BRU as well, which is in June.